In April 2018, citrus canker was detected in the Northern Territory. Posing a significant threat to the Australian citrus industry and trade, the National Citrus Canker Eradication Response was initiated. So what is citrus canker? Citrus canker is a highly contagious bacterial disease that affects citrus plants and some native plant species. It can spread over short distances via windblown rain and longer distances by human-assisted movement through infected plant material and equipment, as well as natural pathways such as insects, floods, severe storms and cyclones. It infects all above-ground parts of susceptible host plants through normal openings and wounds on the plant. Lesions first appear as tiny spots that form into raised corky pustules or blisters with a yellow halo. Although fruit can still be edible, it causes significant impacts to fruit and leaf quality and yield, ultimately causing dieback. Citrus canker caused significant economic losses to citrus industries globally due to damage to crops, minimised fruit production, restricted exporting and increased management costs. The initial detections of citrus canker in the NT were near Darwin Airport and Palmerston. This grew to 16 premises with infected host plants. Encapsulating those infected premises, restricted areas were established. Restricted areas are a 600 metre area around an infected premise that is subject to intense surveillance and movement controls. There were a total of 13 restricted areas within the top end. In each of these, citrus canker host plants could not be kept or grown until the restricted area status was lifted. However, citrus fruit and juice from outside could be brought into the area for human consumption. Control areas were established around the restricted areas. These control areas were put in place to reduce the likelihood of citrus canker spreading beyond the restricted areas. Although the restricted areas have now been lifted, there are still controlled areas in place, requiring permits for any citrus canker host plants or material to be moved out of these areas. The control areas will be lifted upon proof of freedom. Tracing is a process of identifying and locating all potentially infected plants through desktop research and investigations. It assists in discovering sources of infection, minimising the spread of infection, and determines surveillance procedures. Surveillance is undertaken on properties with potentially infected plants. This is to locate trace plants and inspect any other host plants on the property. When a host plant is suspected of having citrus canker, a sample will be taken for diagnostic testing to confirm or deny the presence of citrus canker. All diagnostic procedures are completed under strict biosecurity protocols. It is a requirement under the Plant Health Act 2008 that all potential host plants in restricted areas are removed. Surveillance officers undertake the removal, and this may include cutting the trunk and applying herbicide to the stump to prevent any plant regrowth. Due to the highly contagious nature of the citrus canker disease, decontamination of tools, shoes, vehicles and equipment need particular attention. Strict biosecurity disposal protocols are in place to decrease the risk of spread and contamination of the citrus canker disease. This includes double bagging in biohazard bags, deep burial at an approved quarantine site, or on-site deep burial if required and possible. This also involves monthly inspections of the deep burial sites to ensure safety and compliance with biosecurity protocols. The program has worked closely with the NT Farmers Association, citrus and kaffir lime growers, as well as production nurseries. As movement protocols were established early in the response, inspections by surveillance officers were conducted at least once a week. Within 3 months, approximately 8,600 pieces of fruit and 33,000 kaffir lime leaves had been inspected. The aim was to support the industry and ensure growers would be able to return to trading as quickly and safely as possible. Compliance officers are also working with nurseries and retailers to introduce a monitoring system for restocking plants to improve traceability. The scientific staff have continued their routine testing and analysis throughout the program. In addition to their diagnostic work, there have also been 2 inoculation experiments conducted, one at the Elizabeth MacArthur Agricultural Institute in New South Wales and one at the Berrima precinct in the NT. These experiments will assist in establishing the absence of citrus canker in the NT and will inform and assist with future management of citrus canker. We are now in the final stage of the response and are working on the proof of freedom. This will demonstrate evidence of eradication of citrus canker from the Northern Territory and Australia. This includes significant data validation, auditing and cleansing, which will then be analysed. By the end of 2020, the Northern Territory should be citrus canker free.